Hello everyone and welcome to Blood and Hematology YouTube channel. This video is about morphology of neutrophilic maturation sequence. The neutrophil undergoes 6 morphologically identifiable stages in the process of maturation. These stages include myeloblast, promyelocyte, myelocyte, metamyelocyte, band and segmented neutrophil. During the maturation process, obvious progressive changes occur in the nucleus. The nucleoli disappear, the chromatin condenses, and the once round nuclear mass indents and eventually segments. These nuclear changes are accompanied by distinct cytoplasmic changes. The scanty, non-granular, basophilic cytoplasm of the earliest stage is gradually replaced by pink to neutral staining granular cytoplasm in the mature differentiated stage. The myeloblast is the earliest morphologically recognizable neutrophil precursor. The myeloblast size varies from 14 to 20 micrometers in diameter with a high nucleus to cytoplasm ratio. The cytoplasm is deep blue to light blue in color, non-granular, or contains few granules. The nucleus is usually round or oval and contains a fine chromatin. 1 to 5 nucleoli are visible. Myeloblasts are morphologically divided into 3 types. Type 1, non-granular, with a fine chromatin pattern. Type 2, contains up to 20 azurophilic granules. And Type 3, contains few granules, and features of a blast cell, but lacks a distinct Golgi zone. Promyelocyte is relatively larger than the myeloblast. The basophilic cytoplasm is similar to that of the myeloblast but is differentiated by the presence of prominent reddish-purple primary granules. The nucleus is round to oval, often eccentric, and is still quite large. The nuclear chromatin structure, although coarser than that of the myeloblast, is still open and rather lacy, staining purple to dark blue. 1 to 3 nucleoli can be seen but may be obscured by the granules. The myelocyte is the latest stage capable of cell division. The cytoplasm is slightly basophilic, to cream-colored, and contains few to moderate primary granules and variable numbers of specific, or secondary, neutrophil granules. Secondary granules become predominant as cell matures. The nucleus can be round, oval, slightly eccentric and flattened on one side, or slightly indented. The nucleus is reduced in size due to nuclear chromatin condensation. The chromatin appears more darkly stained than the chromatin of the promyelocyte. Myelocytes are sometimes divided into early and late myelocytes. The early myelocyte has a fine, dispersed chromatin pattern, many azurophilic granules, and few specific granules. The early myelocyte has a more basophilic cytoplasm than late myelocyte. The late myelocyte has a more condensed chromatin pattern, a cytoplasm well filled with specific granules, and rather few azurophilic granules. Metamyelocyte. The cytoplasm is neutral pink color to colorless, with a predominance of secondary granules. The cytoplasm contains very little RNA and therefore little or no basophilia. The nucleus is indented, kidney bean-shaped or peanut-shaped. Indentation is less than 50% of the width of a hypothetical round nucleus. The nuclear chromatin is coarse and clumped and stains dark purple. Nucleoli are absent. The neutrophilic band, or stab form, also called non-segmented neutrophil, is the first stage that normally is found in the peripheral blood. The band neutrophil is slightly smaller in diameter than the metamyelocyte. The cytoplasm appears pale pink to colorless. The nuclear indentation exceeds 1/2 the diameter of the nucleus, but actual segmentation has not yet occurred. The indentation gives the nucleus a horseshoe shape. The chromatin is coarse and clumped. Sometimes in monocytes, the nucleus is horseshoe-shaped and similar to the nucleus of neutrophilic bands. Therefore, band form should not be confused with monocyte. Segmented neutrophil. Although similar in size to the band form, the segmented neutrophil is recognized by a segmented nucleus with 2 or more lobes, connected by a thin thread-like nuclear filament. Most neutrophils have 3 to 4 nuclear lobes, but a range of 2 to 5 lobes is possible. The chromatin is condensed and stains a deep purple-black. The cytoplasm of the mature neutrophil stains a pinkish to clear color. Thank you for watching this video. If you liked this video, to give it a thumbs up and share it. Do not forget to subscribe to the channel, if you still have not. You can click on the bell to receive notifications whenever a new video is uploaded.